Next up, we have Chad Hall, who's a principal scientist at Catalent. He develops technologies for protein expression and manufacturing, including the GPEx Lightning cell line development platform, which he'll talk about. His prior experience in drug development includes at Big Pharma and small biotechs. Welcome, Chad.
Thank you. So I'd like to thank the organizers for the opportunity to present some of our more recent data on developing a GPEx Lightning CHO expression platform for cell line development. We're a commercial CDMO. We play in all the biologic space, really. I'm going to focus today on the CHO cell line development, but we also do vector development, viral vector, cell therapies, et cetera. Just a quick overview, so I'll give some background on the GPEx Lightning technology, tell you about a concept we've referred to as digital expression, and then some examples of how we use this digital expression to really optimize the expression of complex molecules. And then tell you a little bit about, is there an echo? Maybe it's just up here. How we use transient expression for informing our development process. So I'm stealing a review figure here from Boehringer Ingelheim that very nicely shows the commonly used cell line development technologies. So on the bottom left, you have random integration, which is still fairly commonly used. That puts usually concatemers in a few locations, a pretty high copy number often. On the upper is a semi-targeted integration, which a lot of folks use with transposase. And our original GPEx technology fits in this bucket too. We use retrovectors to insert the gene of interest in the CHO genome. And then on the right is a site-specific integration where you usually have a low number of copies, but targeted integration into a very hot locus. So the technology I'll tell you about today, GPEx Lightning, kind of sits between the semi-targeted and targeted. So it's targeted integration into a high number of docks. So we get the high copy number benefits of the semi-targeted, but the target integration aspects of the site-specific. So as I mentioned, our original GPEx technology uses retrovector to insert in active regions of the genome. We can get very high copy numbers from this, very high producing pools from this. So when we developed GPEx Lightning, we wanted to leverage our experience with that technology, and we used the retrovector to insert dock sites or landing pads into the genome. And we also utilized the GS knockout technology from Sigma. So we end up with our parental cell line that has over 100 insertion sites in the genome scattered throughout the genome. And then we can use that parental cell to then go in and insert our gene of interest through a simple transfection. So I'll go through a little bit more detail of that. So as I mentioned, the parental cell line has all these dock sites, and in that dock site is a recognition sequence for the 5C31 recombinase. So that there's a recognition sequence on our plasmid of interest too that has the selectable marker in the gene of interest. So the recombinase mediates the insertion of the entire plasmid into the dock sites of the genome. And this is done very quickly in a transfection. It's a really quick reaction. Of note here, I'm showing some primers in red and green. One primer sits in the dock site, one sits in our boat plasmid sites. We refer to our plasmids as boats because of the dock analogy. So upon successful insertion, it creates a unique amplicon so we can quantify copy number just simply based on qPCR. And I'll mention here our GS selection that we use. We played the tricks that a lot of other folks use of really hindering the GS expression so that pools need a lot of copies of the expression cassette to survive selection. So here's the performance of a sample of antibodies in a fusion protein. This is cells are transfected, glutamine's removed from the media, and they die off a little bit and then come back. Usually takes about 12 to 18 days for recovery. We're actually working on speeding up that process to shorten timelines. So after selection's done, the integrated copy number using that amplicon assay I showed in the previous slide is quantified. So pools, typically, this is older data now, this is 30 to 60 or so copies in the pool. We're doing a little better than that now, up to 100 copies in the pool. And these are pretty rich pools in terms of copy number and titer. So here, this is kind of displaying the consistency of the system. So this is 3 replicate pools. So 3 different transfections for a mAb antibody. Pools are selected and then put into production. This is a generic fed batch production of the pool. You can see the VCDs track very nicely, very consistent viabilities and titer, all pretty spot on. So that actually allows us to go from 3 replicates to 2 as a backup so that we can test more samples. So here, now I'm switching gears to the fusion antibody. So this is the first set of clones we got out of Lightning. And we saw very good correlation between the percent dock fill, so the number of inserts, and the titer. So it's a nice linear relationship, so we can screen clones based on qPCR, since the gene of interest is going to a consistent number of dock sites and consistent locations, it's a predictable output. So more copies equals more titer. The clones from these pools are also very stable. We have very good stability with both our GPEX technology and the new Lightning technology. The process is also very scalable. This is actually the first Lightning project we put into manufacturing suites. You can see the 2-liter, across the 2-liter, 50-liter, and 500-liter subs. We had very consistent VCDs, viabilities, and titers. This client actually wanted to reuse an old column, so they were able to shorten the timeline for their productivity down to 14 days, because we actually would have made too much titer for their column, so we backed it down, which is a nice luxury to have. We typically see very high and prolonged viabilities, allowing us to extend the production culture out if we need to get more product. So that's kind of a background on Lightning. So now I want to talk about a concept of digital expression. I'll explain what that is. So the traditional vector design shown here on the left, which I'll refer to as analog expression, I'm preaching to the choir here that the better you can get your product quality and your titer and your cell line development campaign, the better the upstream, downstream purification, the better life is in general, right? The more you can fine tune the process, especially for like bispecifics, the better heterodimer formation you can get, the better off you are. So for a traditional process, you'll play with the vector design. You'll switch the orders of the, in this example, heavy and light chain, but it could be a variety of different genes, a number of genes. You'll play with promoters and introns and a lot of different genetic elements to find the right balance of expression. And that may vary project to project because of differences in translatability, et cetera. So it's a lot of work. We've all suffered through this some, especially if you're in the molecular biology team. So we wanted to leverage our very high copy number to see if we could use that to adjust expression. So the concept here is that we'll put the heavy and light chain on 2 separate plasmids and transfect them together and at different ratios. So instead of a 1-to-1, we can do a 10-to-1 or whatever ratio we want at transfection, so that the copies inserted will express different amounts. If you have twice as many heavy chain copies, you'll get twice the expression. So I'll walk through some examples of how we use this concept to really get as good a product quality and titer out of the pools before we go into the cloning stage. So we like to do a lot of our initial characterization of the pools. So here's a bit of a busy graph here on the left. So what we did for this experiment, this is a simple mAb example. We made a series of pools on the x-axis here, the leftmost that had 10% light chain plasmid and 90% heavy chain plasmid. So as you go across the x-axis, you go all the way to the opposite, where it's 90% heavy chain, or sorry, 90% light chain, 10% heavy chain. And on the left-hand y-axis is the integrated copy number of heavy and light chain. And you can see as we change the ratio in transfection, we get the expected, or hoped for at least, change in the integrated copy number. So it tracks very nicely And when we run these in a generic fed batch productivity, you can see that titer is maximal at that 50-50 range, which we all know from transient work is what we kind of expect there. But what's, I think, very interesting is not only can we look at titer, these different ratioed pools, but we can look at product quality. So the right-hand side is a page gel of unpurified soup. So we like to look at unpurified supernatant to see what, so we're not blinded to what might be lost in the purification process. So on the left here, the 10% light chain pool, so 90% heavy chain. You can see the fully formed product at the 150 kilodalton there, but also some excess heavy chain, some aggregate up top, which might be expected when you're overexpressing heavy chain. And as you go from left to right, you can see in the 50-50 range is where we're getting the optimal product quality of just a little bit excess light chain or light chain dimer, and mostly fully formed antibody. And then on the right, you can see that we have a lot of excess light chain, both monomer and dimer, which is really just leaving titer on the table. The cells are producing the light chain. It's not making it into your final product, so that's not a desirable state. So the system really allows us to control expression in a simple ratioing manner at transfection. So the next question, so that's pool data. How do the clones look? What kind of variability do we see in the clones? So I'm showing here a series of digital expression pools. These are all monoclonal antibodies that were made at a 1-to-1 heavy to light ratio. This is the portion of light chain in the clone. So box plots there of the clone distribution of the light chain portion. And you can see some variability project to project, but all around that 50-50 mark. And on the far left there is actually the control of a bicistronic vector that has both heavy and light chain. So that is, by definition, a genetic 1-to-1, but it still shows some variability. So this probably just reflects the variability of our qPCR assay. And my kids are very happy that I included this reference to Gassman and Horn of 2012, that the lightning cells get what they get, or take what they get, and they don't throw a fit. So what we give them in the transfection mix is what they insert. So it's a pretty robust system. So I'll walk through some examples of how we use this system. So this was a project where we were expressing a very asymmetric knob and hole bispecific, asymmetric in terms of size, but also in terms of expression. The antibody portion of this expresses very well the FC portion. The fusion portion does not, so we knew we'd need to balance things quite a bit here. So in the series of pools we made, we started at 5% of the MAB, 95% of the FC, and you can see the portions we varied down to 50-50. And the integrated copies is shown next to that what we gave them there, again, is what they get. So it was a predictable outcome. So we, obviously for bispecific here, product quality is of a large importance. So we run a page gel on these pools. I'll mention on the reference standard on this gel on the left contains both the purified MAB as well as the purified fusion dimers as references there. So as you go for the 5, 10, 15%, you can see you're decreasing the amount of excess fusion protein. Let's see if I can point. The fusion protein here is going down while the desired heterodimer is going up, and also the fusion monomer is going down. And as you go up to 30% of your MAB, you're seeing the MAB impurities come through the homodimer of the MAB and the half MABs. So we can really fine-tune where the proper ratio is in these pools. And shown on the right here is the capillary electrophoresis of the same thing. I won't go through that. Here's another engineered bispecific knob and hole FC fusion. There was a bit of a challenge. I won't go through the ratio detail, but we identified the optimal ratio and got pretty good titer and expression and growth profiles from this. So generic fed batch, we got over 8 grams per liter and very robust viability and growth through the process. So this example is another very challenging example, this product required a protease cleavage to activate the molecule. The problem is the protease was known to be deleterious to CHO cells. They don't like expressing it. And it's also promiscuous. It can overcleave the product. So there were some concerns here. We knew we'd really need to balance expression of the protease compared to the product. So we did a pretty drastic range of ratios. So at the 1-to-1 ratio, where we have a lot of the protease, you can see right off the bat that we got few copy numbers in our pools, which indicates that the molecule is toxic, which we expected, and the product was overprocessed, so overcleaved and inactive. When we go to the 1-to-10 ratio of enzyme to product, we get a few more copies in, but also the molecule's overprocessed. When we went to the 1-to-100 ratio, so 1% of the enzyme, 99% or so of the product, we saw the optimal processing, where it was fully processed in the desired cleavage spots, but not in the promiscuous spots. And as a control here, I'll mention, I mislabeled this. I noticed this morning, this is pure product with no enzyme, so it's a 0-to-1, not a 1-to-0. We get high copy number like we'd expect of 84 copies that we saw with a small amount of the enzyme present. And that is unprocessed since it doesn't have the enzyme. So from this, we could see that 1-to-100 pool was really where we wanted to be in terms of product quality and in terms of titer as well, which I'm not showing here. So we cloned this pool and analyzed it for the copy numbers of the product and enzyme. And you can see this pool had on average 0.3 copies per cell of the enzyme. So as we might expect, it was actually comprised of clones that did not have any copies on the right of the graph here. And then some clones with 1 copy of the enzyme, some clones with 2 copies of the enzyme. So that gives us a really high dynamic range of expression, because we can get all the way to 1-to-100 or so ratio of expression without having to do cloning. So this would be a very challenging molecule to express with traditional vectors to get that kind of a difference in expression. So now I'll tell a little bit about our transient and how we use transient expression to inform the development process. So there's really 2 purposes for transient expression as we see it. One for bulk protein production. We can do that with our system, but we rarely do, because we can go through selection fairly quickly and get high producing pools. So we don't really have a huge need for transient expression for bulk protein production. But what we see about a value in transient expression is by testing ratios quickly to scout to find the best ratio for the pools and to select which pools to take into clonal. And I'll walk through that data. And I should also mention, I think I forget to say this at the beginning of the talk, but this cell line is available for out licensing. So you can test the cell line and the processes in your lab and use it for earlier phase candidate selection, stuff like that. So here, I apologize for the ugly gel, but we can still conclude from it. So what we do is we do the pool transfection over a range of ratios of heavy to light chain. This is the same example I showed earlier. After the transfection for the stable pools, we split off some of the cells to do transient productivity. So this is the exact same cell population that goes into the stable pool production. We split that off, do a 7-day transient production, run a gel on this, and I'll mention that the gel here is loaded based on constant volume of the supernatant, not on constant mass of the mAb or titer. And as you can see, you see the same pattern on the left-hand side. When you have excess heavy chain copy numbers, you see those product-related impurities, and notably the 150 kilodalton chain is less prominent than it is in the middle, the 50-50. So this tells us kind of what we knew, that we can use this to scout, or that the 50-50 range is the optimal place to be for this mAb, but that we can use these transient early reads just a week after the transfection process to figure out which pools to take forward, and which, not only for production and freezing, but also for clonal. So on the right-hand side is the stable pools. So after the pools are selected, put through normal generic fed batch, you see the same pattern we saw before, where the left-hand side you have too much heavy, right-hand side you have too much light. So we were really happy with the alignment that we saw there. And maybe that's not surprising. These are the exact same cells. This is not a separate transient expression cell line. It's really the true CLD parental. So in terms of product quality, or of titer with our transient system, can we use that to predict which pool is going to give us the best titer? I'm showing here, this is two different medias for both the stable, so the left-hand side of the graph is the transient expression, cell-specific productivity, the same range of pools here for the percent light chain at transfection. And on the right-hand side of the graph is the stable pool, so you see a nice correlation between both medias actually with how the transients perform and the stables perform. And the same for volumetric productivity, that the transients really telling us which ratio and which expression level is optimal for titer as well. So just to conclude here, we feel the GPEx cell line offers a lot of benefits, very quick development timelines to be able to optimize expression quickly and get early reads on product quality and titer. So our selection process is very robust. We get a lot of copies. Clones with up to 100% dock fill are not uncommon. We have tunable expressions, so we can really get that product quality right early. Very high titer. I think our average monoclonal antibody project right now, the top clone is in the 9 gram per liter range. And I think up to 18 grams per liter for the highest titer we've seen out of a mAb clone. Bispecifics, we can get 11 grams per liter, which is also comes with good product quality there. All of our clones are very stable. The platform's scalable. We have a good track record with GPEx as our kind of backbone here. 90 clinical products and 20 commercial. So where we stand on Lightning, so we launched this system in 2021, this digital expression as a more recent development of it. We started out with a normal bisystronic vector. We've done a variety of molecules, and we tend to attract clients that have very interesting designs on their molecules, and we're pretty good at executing on that. So we've done a total of 204 projects, 161 for clients, and 43 internal to develop the technology, and also to pair it with our smart tag targeted ADC, targeted conjugation technology with the Lightning system. So our site at Madison obviously does cell line development, but we could go all the way through GMP manufacturing with all the necessary analytics and process development groups attached to that. And again, this cell line, this technology is available for out-licensing, both R&D scale and commercial scale, if it's something you'd like to try out. We have a booth 718, or find me afterwards if you want to chat more about it. But happy to take any questions. Thank you.
Thank you, Chad, for introducing in very detailed manner your GPEx Lightning system. We have about 6 minutes-ish for questions before we go on break.
Hi, thank you. That was really great. I have a question about multi-specifics and light chain mispairing. Have you guys analyzed your platform for that?
Light chain mispairing for bispecifics?
Bispecifics, try multi-specifics.
Yes, yeah, that's something we're definitely looking at. I'm not as familiar with that data, so I'm not sure. But we can also balance, so when different light chains, we can balance up to, I think we put in 5 different genes, so we can get pretty tricky with all the ratios of heavy to light to make sure things are balanced in terms of expression. Pairing's obviously a different beast, but yeah.
How many pools would you make for a typical project with a multi-specific like that? Because there are so many different ratios you could do.
Yeah, exactly. So usually the clients have some sort of idea from transient work where the ratios need to be, so we usually start around there and kind of blow it out. And lots of times that's up to the client, what their appetite is for the complexity there. And it is, since we can get the read from the transient pretty quickly, we can, if we need to, go back and restart within 2 weeks for sure to fix any misses we had on what the proper ratio was. But we tend to cast a wide net so that we don't have to do that.
Sorry, then last question. With hundreds of copies, well, up to 100 copies, do you guys notice any issues with stability of your clones?
No, we see very, very good stability in clones. It's a little bit surprising how stable things are. And actually, we see good stability usually in pools, which is even more surprising. So, yeah, no, it's a very, very stable system. And that kind of harkens back to the GPEX system was also very robust stability. So,
thank you.
Hi, Chad. Thanks for the really nice presentation. So I had a question about the ratios, but that's been already asked. I assume for a 3-chain or a 4-chain molecule, this optimization can get quite complex. My question about when you move forward to clone selection, let's say you optimize the ratio, but among the single clones, I assume there will be a distribution of copy numbers with the mean being your target.
Sure.
So how difficult it is to then pick out the best clone in the next step?
Actually, I had a slide related to that. So this is the clone distribution here for the MAB2 project. We were shooting for 50-50, and the clones range from 40 to 60. A large portion of that is probably variability in the qPCR assay. So our clones tend to match the pool very, very nicely, so that that's both a good and a bad thing, because you can't fix problems in clonal, but you already have the pool that fixed the problem most likely. So once we know the transient data and what pools are in the right range, we can take multiple pools into clonal and narrow things from there. But in general, the clones look just like the pools in terms of the genetics and the product quality and glycosylation, et cetera.
Cool. Thank you.
Thank you.
Thanks. I may miss this. How you have identified your locations for all these integration sites?
Sure. So we didn't. So we let the virus tell us where the hotspots are, and that's kind of what GPEX does. It's like the transposase technologies. They go into the hotspots. So, and actually that brings up a good kind of side point that surely different dock sites are hotter than others, but the sheer number of dock sites, I think, evens all that out. So I think that's why we see good correlation between copy number and titer. It matters less whether you're in a hot dock or a cold dock, because there's 100 copies there. So it kind of averages things out better.
Very nice talk. A question about the dock fill. So usually for one project, how many clones do you screen for the dock fill? And what's the cause the difference for the efficiency?
So maybe I didn't mention, we use the Bruker's Beacon platform to screen clones. So we get a read on titer on that instrument early on, and then we export top 96 clones and take 24 into Amber screening. Does that answer your question? I'm not sure if I... Okay.
Here you go, sir. Yeah, what causes the difference of the dock fill efficiency?
What affects the differences in dock fill efficiency?
Yeah.
Yeah, so the data I showed was older data, so we're much more consistent in our dock fill. We're in the 75 to 100 copy range typically now for pools. I don't know the exact answer to that. Why isn't it 100%? I don't know, but it typically is in that 50 to 75% fill range, independent of how many dock sites you have. So it's yeah, not sure why it's sometimes a little more, but by the time we go through clonal, we're typically in the 80+, 90+% fill, so it gets consistent in clonal.